glasses of wine are all filled to go through another complex talk at that point of time after many interesting talks. So today I would like to present you some data how we can come from a facial picture to a mask that can be used to identify a gene that is mutated in this patient in a pathway disease. So I'll just quickly introduce you to this pathway disorder. It's called GPI anchor deficiency. And the GPI anchor is basically a sugar molecule present on every cell in the human body, and it links hundreds of different GPI anchored proteins, and it is constructed, or in the synthesis of this anchor, there are 29 genes involved. So 14 of these genes involved in the synthesis of the anchor are already linked to diseases. And I would like now to focus on 5 of these genes. So just to quickly explain you that on the normal cell there are lots of GPI anchored molecules present. In a GPI anchor deficient patient, for this case a patient with a PIC-N mutation, there are only few GPI anchored molecules present, and thus GPI anchored proteins on the surface of these cells can be measured by flow cytometry, and you can see a reduction in different markers. However, this reduction that we see is individually so different that in patients with another mutation, for instance PJP3, the reduction is also present, but it's variable, and it is variable from individual to individual. You cannot see a common pattern between patients that have a mutation in one gene and another pattern with mutations in another gene. Therefore, we are looking to find a way how clinicians can diagnose these gene mutations in patients by looking at their faces. So we will focus now on these 5 genes, and what we have done is we collected facial pictures of 52 patients from all these harboring mutations in these 5 genes of the GPI anchor pathway, and we constructed these, with the help of Face2Gene, these syndrome masks. In total we used 68 images and 52 patients. So we then performed a cluster analysis and a confusion matrix. So in a totally heterogeneous disease, the probability that a gene would be mapped to a correct or a false positive that a facial picture would be mapped to a gene, to correct gene, or to false gene would be 0.2. So it's 1 divided by 52 cases. In a completely monogenic disease, you would align all the true positive hits on a diagonal. So we performed these analyses now with the data, and what we can see is that we have a diagonally aligned true positive hit rate of between 40 and 80%, and the mean accuracy is significantly higher than a random guess with a 51.55%. What you can also see is that there are high false positive rates in the red and green boxes, and they basically cluster for a common substructure of the disease. And these are details that I don't want to go into much in this small round, but if you want I can explain you at my poster with number P0820D tomorrow. So I'll be very happy if you would meet me there and have a chat about GPI anchor deficiency and how Face2Gene helped us to identify genes mutated in patients with a pathway disorder. So thank you very much for your attention.